Bio. This is Mr. B. We're continuing on with Section 1. So this is Section 1, Part 2, the work of Gregor Mendel. So we're going to jump right into it here with our first question that we're going to ask in this section is, what is the principle of dominance? So that we're just going to basically ask, what does it mean when we think of dominance in terms of genetics? So the principle of dominance states that some alleles are dominant and others are recessive. So remember we said allele is just a different part of a gene. Remember we said you can either be tall or short, so we have a tall allele and a short allele. So some are dominant and others are recessive, meaning dominant ones will always be shown over recessive. And an organism with a dominant allele for a trait will always exhibit that form of the trait, which I just said. So we can have, if we have the allele for being tall, we're going to be tall. Or I guess in pea plants, if they have that allele for being tall, they're going to be tall. It doesn't matter if they have one tall, one short, or two talls, they're always going to be tall. And an organism with a recessive allele for a trait will exhibit that form only when the dominant allele for the trait is not present. So that just means that, again, what I just said, even if, so there's two alleles for each gene, so tall and short, it would have to be two short alleles in order for the pea plant to be short. They can't just they will never be short if the tall is present. And we're going to look at what happens during segregation. So segregation is kind of another part that kind of goes along with dominance. So just know that the difference between a dominant allele and a recessive allele. And what happens during segregation. So segregation is going to be a big term here. So when we think of segregation, it's just going to be like separating. So we're kind of going back maybe to history, you've heard about segregation occurring, just separating the different races. So now we're going to look at separating different genes and alleles. So how Mendel discovered segregation was he crossed the F1 generation with itself to produce the F2 generation. So just know, don't worry about the filial part, just know that we have F1 and F2. And the traits that were controlled by recessive alleles reappeared in 1/4 of the F2 plants. So we're going to see a picture of this and it'll make more sense. So here is kind of what this is saying. So we have our parent generation. We have one tall, one short. He mated those together and he got two tall plants. So that's kind of what they expected. Since the tall allele was present, we're going to get two tall plants. However, once we took the two tall plants and mated them together, we actually got one short plant finally. So we had three talls and one short. So that short allele was kind of masked, but it kind of came back into play and showed up in the F2 generation. And Mendel assumed, so after looking at these results here, getting his one short plant, he assumed that a dominant allele had masked the corresponding recessive allele in the F1 generation. So basically what he meant was, in our F1 generation right here, that recessive allele is present in both of those plants, however, it's just masked, and when they mated, they came out with they were able to pass on that short allele to that one plant, and that is why it is short. And the trait controlled by the recessive allele showed up in some of the F2 plants. Again, that's what we just said. And the reappearance of this trait controlled by the recessive allele indicated that at some point the allele for shortness had been separated or segregated from the allele for tallness. So I think I said that segregated just means separated, so that's just showing that even though they're the same gene, they can be separated from one another. It doesn't matter that they are kind of connected on the same gene, they can still separate. And so going along with this kind of concept here, Mendel suggested that the alleles for tallness and shortness in the F1 plants segregated from each other during the formation of the sex cells, or gametes. So gamete is going to be a term we're going to hear a lot. And that's just what the terms for our sex cells are, the sperm and the egg. It's just a fancy way to say that would be gametes. And when each F1 plant flowers and produces gametes, the two alleles segregate from each other so that each gamete carries only a single copy of each gene. So we're going to talk about how our sex cells divide. They divide different than our regular body cells. And but what it's saying is that our sperm will only have one allele for tallness. So the flower sperm only had one allele for tallness, and then maybe has one allele for shortness. So therefore, each F1 plant produces two types of gametes, those with the allele for tallness and those with the allele for shortness. Again, what I just said. And this is what it kind of looks like here. So our first generation, you can see it had the big T and the little t, so tall, short. And then segregation occurs, so now we have 4 separate cells here, which we're going to talk about later how this happens. These are in our gametes, and then when they come together, they give us all these different combinations. So that is it for that first section in this video.